and I want to introduce the interim dean, Dr. Brent Nelson, to come give some opening remarks.
Thank you very much. So good evening, everyone. I am Brent Nelson. I'm the interim dean of the College of Science. And it's my pleasure to welcome you to the extraordinary celebration of undergraduate research in the College of Science. So, to our students, faculty advisors, parents, families, and especially our generous donors, thank you for being here tonight. What you're about to witness is nothing short of remarkable. The brilliant minds of our undergraduate researchers, sharing discoveries that advance knowledge, solve real-world problems, and demonstrate the transformative power of hands-on scientific inquiry. Undergraduate research is not simply an enhancement to classroom learning. It is essential to developing the next generation of scientists, innovators, and leaders. When our students work alongside faculty on frontier research, they don't just learn about science, they become scientists. They develop critical thinking skills, learn to navigate failure and breakthrough, and gain confidence that will serve them throughout their careers. These experiences often shape the entire trajectory of a student's life, opening doors to graduate programs, career opportunities, and a lifelong passion for discovery. But here's the truth. None of this happens without you. The research you'll see presented tonight exists because of the generosity and vision of donors who believe in our students and our mission. Philanthropy creates opportunities that would otherwise be impossible. Philanthropy provides the scholarships and fellowships that allow students to dedicate their time and talent to research, to pursue their intellectual curiosity and reach their full potential. I want to emphasize something important. Gifts of all size make a difference. Whether it's $50 or $5 million, every contribution matters. Let me share for you an inspiring example. An alumna who graduated in 1948 put herself through school with little resources and was profoundly grateful for her Northeastern education. She began giving back in 1969 with minimal funds. And over the years, her commitment grew. In 2012, she and her husband created a fund to support undergraduate students in co-op research positions. Through a planned gift, their fund was recently endowed with a $1 million gift from their estate. Endowments provide permanent, sustainable support for generations of students to come, ensuring our programs can thrive not just today, but for decades into the future. This fund has already changed lives, and it will continue to change lives for generations. This is the profound and lasting power of philanthropy. And while the above story example exemplifies transformational philanthropy, I want to equally emphasize the critical importance of the gifts we receive for the Dean's Fund. This fund comprises gifts from parents, students, alumni, and friends, and some faculty and staff. And it's the reason my team can say yes to students seeking these valuable summer experiences. Tonight's scholars have been supported through individual scholarship funds created by donors in this room. I'd like to give special thanks to the Richard W. and Elaine E. Barrett Foundation, the Halberstadt family, Russell Women in Science, and Professor Paul and Irene Voros. Your investment in these young scientists right here validate their hard work, demonstrates that their community believes in them, and provides the practical support they need to excel. I also want to express my deep gratitude to Rochelle Reisberg. I'm not sure if Rochelle's here tonight, but her exceptional team was critical in organizing tonight's event. Rochelle's leadership of the Dean's Undergraduate Scholars Program has been nothing short of transformational, and I'm deeply grateful for her effort. This program provides structure, mentorship, and community for our undergraduate researchers, and it's become absolutely essential to our college's mission. The care and dedication that Rochelle and her team bring to supporting our students is extraordinary, and we are fortunate to have such committed advocates for undergraduate research. So as you listen to tonight's presentations, you'll quickly discover the passion, intelligence, and potential of these remarkable scholars. I'm going to add humor, so I hope you deliver. And I hope you feel, as I do, immense pride in what we're building together here at Northeastern's College of Science. Your presence here tonight, whether as a supporter, a family member, a mentor, or a faculty member, makes this possible. Thank you for investing in excellence. Thank you for believing in our students. And thank you for helping us create a future where undergraduate research continues to thrive at Northeastern. Thank you very much. Right, at this time, I think we're going to begin the student presentations. But I do not know who the first speaker is. All right, we have a volunteer. Excellent. Great. Oh, it's Benjamin Wasserman, who I'm happy to introduce to you. I guess you're going to tell us a little bit more about yourself. I don't want to steal your thunder.
Good evening, everyone. My name is Benjamin Wasserman. I am a fourth-year psychology major with minors in nutrition and behavioral neuroscience. So beyond academics, I am the undergraduate coordinator for a Northeastern Psychologists for Inclusion and Equity Committee, along with a peer supporter with Lean On Me, which is a text-based mental health hotline available for Northeastern students. So my primary research interests lie in health psychology and behavioral medicine, with a particular focus in positive psychological and mind-body interventions and their impact on stress physiology, immune function, and overall health outcomes and wellness. So these interests ultimately led me to Massachusetts General Hospital's Center for Neurointestinal Health under the mentorship of Dr. Helen Burton Murray. So since starting back in January, I'm so grateful to have had the opportunity to contribute to 2 abstract submissions to conferences, including one titled Disrupted Interoceptive Awareness in Functional Dyspepsia and its associations with symptom severity, and the second being limited diagnostic concordance between ARFID screenings and provider documentation in a neurogastroenterology population. In addition to that, I've had a really meaningful experience developing my own independent project, which is currently in the data collection phase, exploring spirituality and resilience in patients with functional dyspepsia. So, how did this abstract research project really come about? Well, at MGH Neurogastro, we study what is called disorders of gut-brain interaction. And what those are, they're GI conditions that present without structural or systemic abnormality, so there's really no physical cause. And one of these conditions is called functional dyspepsia, which is associated with abdominal pain and bloating, nausea, to name a few symptoms. So what we do know from empirical studies is that there is a strong connection between the gut and the brain. And if you look at the figure, it's a little small, but in the bottom left is what we call the gut-brain axis. And to explain it in simple terms is alterations in our gut can alter our mood, our behavior, and even our mental health. And this is a bidirectional relationship. So what that means is that stressors from our daily lives or our emotions can also influence gut motility, secretion, and overall microbial balance. So as a psychology student, I was super interested in this because I wanted to know how could mental health influence GI symptoms. And at this time, while I was at MGH, I came across a cutting-edge fMRI study that found that spirituality can be protective against mental health conditions such as depression and anxiety, while also strengthening resilience pathways in the brain. And when many people think spirituality, they automatically associate it with religion, which is not necessarily the case. So spiritual awareness can be your ability to connect with something greater than the self, a love for the universe, or a sense of peace from within. So knowing this from these studies, I began to wonder if cultivating spiritual awareness in patients with functional dyspepsia decrease symptom severity and improve treatment outcomes. And I also wanted to know if spiritual awareness and resilience differs between functional dyspepsia patients and healthy controls. So I hypothesized going into my project that greater spiritual awareness promotes resilience, which in turn decreases symptom severity by reducing overall GI anxiety and hypervigilance. So to test this hypothesis, I implemented two well-validated measures into our studies. So the first one is titled the FACET SP12, which measures spiritual awareness specifically in chronic illness populations, and then the BRS, which measures resilience, one's ability to bounce back from adversity. And my goal going into this project was to get 10 functional dyspepsia patients and 10 healthy controls at a minimum, and later analyze these findings. And I am very happy to share that we're almost at those 20 participants. So this winter, I will hopefully run an analysis and get to present my findings at a conference later next year. So to conclude, I would like to thank Dr. Helen Burton Murray for her mentorship, my supervisors, Blythe Peterson and Jenna Kluke, for supporting me through everything and encouraging me to do this independent project, which has been so meaningful. And of course, the Paul and Grace Ward Martinez Co-op Fellowship for supporting my research endeavors. Thank you all for listening. I hope you have a beautiful rest of your night. And it is now my pleasure to welcome up Tim Wessling.
Thank you, Ben. Hi, everyone. My name is Tim Wessling. I am a fifth year, aka ancient, astrophysics student. I am one of the co-presidents of the Society of Physics Students. I also sing in madrigal singers from time to time. My main research has been with the Armaki Lab. I've done 3 co-ops working with Sugo Armaki, and most of that has been focused on the astrophysics research that I will talk about on the next slide. I also dabbled in the cosmology group with Professor Jacqueline McCleary. She's great. And I did some work with circumgalactic dust halos. Future plans, I'm sure as many of you in this room are in the depths of applying to PhD students, which as we know this year is very challenging. But I would love to be a tenured faculty someday. Working with students and being a mentor is something that really excites me. And that's me at a Chaperone concert. It was amazing. So now I can talk about my research. So I'm very interested in something that's called a blazar. I'm sure many of you know that we have seen that at the center of all galaxies is what's called a supermassive black hole. I'm sure a lot of you have heard of black holes in pop culture. Some of you maybe have seen Interstellar. It's a great movie. And what's really interesting about black holes is that when material falls really close to them, their gravitational attraction tears that material apart, and that material orbits at very high velocities, and it becomes very warm in what we call an accretion disk. It's through some very theoretical, we'll say, mechanisms, material is funneled into these relativistic jets where charged particles move at very high energies, very high velocities. And what you see on this picture here is you get a relativistic effect called Doppler broadening, or Doppler boosting, my bad, where basically the luminosity is greatly boosted, and you get this really bright source that almost looks like a star. And in fact, when astronomers first observed these sources, they thought they were stars, and they adopted the name quasars, or quasi-stellar objects, for these sources. But they're so unbelievably bright. So the way we study these sources, light is emitted across many wavelengths, from radio, X-ray. You've probably heard of many of these in your daily lives, but we are actually interested in the highest energy emission, gamma rays. Light is emitted from these sources across all wavelengths. So if you look at the figure in the middle top, we can see that light is emitted across all sorts of wavelengths in the kind of 2 humps. The region that I'm interested in is the gamma ray region, the highest energy emission that we have. So we can see in the plot next to that one on the right, that is the spectra of a source in this region. But when you're doing broadband astronomy and astrophysics, you need a collection of data to basically accurately model what the spectral energy distribution looks like. But there's a problem. MEV gamma rays, which is a certain energy of gamma rays, have not been observed very much. This is because there hasn't been a lot of detector technology since the 90s when we had a mission called CompTel to basically get high sensitivity data for this region. So it's very difficult to accurately model the spectra of these types of sources across this region. So what we do is we conduct a cross-match. We take an X-ray catalog. X-ray is slightly lower energy than gamma rays, and then we take a GeV catalog, which is instrument that measures higher energy. MEV is mega, GeV is giga, so it's larger. And we put them together and we say which objects were detected in both. and we can take that to model the spectra, which is what we do, of these sources across the MEV gap. Basically, you get an idea of what the spectra looks like across this whole region, and you can model what's in the middle. We use this to make predictions, like the maps that you can see. And this is really important because we want to motivate MEV science. There's a lot of really exciting missions that are coming up. The one that I work on is called GRAMS, which is the Gamma Ray and Antimatter Survey. There's one planned to launch in 2027 called COSY. That one's going to take a look at really interesting line emissions in this region where a lot of nuclear physics happens. And then E-astrogram is another mission that's going to happen. There's a lot of really interesting physics that happens in this region, especially with non-thermal physics. And we want to understand that so we can understand things like what's dark matter? How do supermassive black holes evolve? We don't even know how supermassive black holes form. It's very theoretical, but they're really interesting. They can tell us how the universe behaves overall. So like I mentioned, there's some glamour shots here. They're very beautiful. And I'm very interested in really learning how the universe evolves, how supermassive black holes evolve, because like I said, they're very mysterious. And this is really what kind of got me into science. In the first place, I was just really interested in the mysteries of the universe, things that we don't really understand. So I'm very excited to say that the work that I've been doing with Sugo's lab is being put into a paper. It's in prep, so I'm hoping to publish that before I apply to PhDs in December. And I'm very lucky that I have an amazing lab to work with. Sugo Armaki is one of the most kind souls I've ever met. He's amazing. He's so kind, and I am lucky to work with a bunch of PhD students. And as you can see from the top right, I'm very lucky to work with collaborators from not only across America, but all the way around the world. I work very closely with a collaborator, Naomi Tsuji, in Japan. She's lovely. We call on Zoom, and it's like a 13-hour time difference. It makes international collaborations quite difficult, but we make it work. And also from NASA, the Goddard Space Flight Center, they're great too. But yeah, I'm really grateful for the AJC Merit Scholarship. I was really honored to be selected by that, and the URF for honoring me with that award. So yeah, I want to thank them. And without further ado, I believe the next speaker is Sarah Perkins. So she can come up now.
Thank you, Tim. Hi, everybody. I'm Sarah Perkins. I'm a third-year ecology and evolutionary biology student here at Northeastern. And ever since I was young, I knew I was passionate about research. And when I got into high school, I knew that's exactly what I wanted to do. So I went straight for cold emailing as many labs as I could, and eventually one took me in a town right next to my hometown of Chelmsford, MA. Shout out to Chelmsford, at UMass Lowell, called the GARB Lab. And there, they worked with spider genetics. But I knew going into university, I wanted to work on something I was much more passionate about, which is ecology. And so I started cold emailing as many labs as I could going into Northeastern, and the Kimbrough lab took me in. And I'll start talking about some of that research today. And being in the Kimbrough lab really jump-started more of the research I've done here at Northeastern, and I'm super thankful to them for that. And actually, you can see me here in rain gear, 7 hours away from here in Lubeck, Maine, collecting snails, predatory snails, for some of my research I did in a different lab at Northeastern called the Trussell Lab. Super awesome. I'm super wet, super soggy, as you are in Maine. All right. Always. So my research in the Kimbrough lab started like this. So before we can actually get into that research, I want to give a little bit of education on what actually happens in the ecosystem we focused on in my research, which is the New England kelp forest ecosystem. So in that ecosystem, we have our secondary consumers, which are your Jonah and your green crabs. They share the same niche, and they work to consume both mussels and urchins in the ocean, in the Atlantic, the very cold Atlantic. And those urchins work to consume sugar kelp, our native sugar kelp, which also produce oxygen and sugar for those urchins and other organisms to eat. So it's a very neat cycle. But the question we were mostly interested in studying in our lab was, if those urchins, if they are bystanders to crab predation, so they witness or they somehow sense these crabs preying on mussels, but they are not preyed upon by those crabs don't have access to those urchins, do those urchins change the way they allocate their energy? So we as humans can allocate our energy in tons of different ways. But for urchins, there's really two main ways we're interested in. So we have like the super successful way, which is reproduction and growth, right? Like that's the gold standard. You want to pass your genes on to the next generation. That's the ideal. And then you have the other way, which is we have some listed here, but the main other way we were interested in was evading predators. So they have a lot of different behaviors that enable them to do that. So how do we actually study that? Well, we have these giant massive tanks. If you ever actually go visit the Kimbrough lab at our Nahant station, the Marine Science Center, you'll see it's super loud. We have these 28 massive black giant, like there's tons of gallons in these tanks. They're almost as tall as I am. They're super loud. They have these seawater pumped in straight from the Atlantic. And they house a few different things. So all of them house urchins. All of them house some kind of kelp. And half of them house crabs and half of them don't as a control. So we know that some of them are being exposed to this crab predation. The crabs are held here in lobster tanks, you can see from my very rudimentary drawing. And those crabs are eating mussels, but they don't have access to the urchins. But how do we actually collect data to evaluate that question? Well, the main way we collect data on how they're evading predators is through their behavior. So one of the cool ways that urchins will evade predators is that they'll decorate. This is a really cool behavior you can see here. They're actually very mobile organisms. A lot of people think of them as sedentary, but they actually do move around a ton. And part of what they'll do is they'll pick up little things in their environment and put them on their spines to sort of camouflage themselves in their environment. So we would go every day and we would measure whether the urchins were doing that. One of the undergraduates when I was working there actually came up with this really cool way to see if the urchins were moving to different levels in the tank over the period of the day, which was awesome. And then, so that was the main way we measured how they were evading their predators. But how do you measure reproduction and growth? It's pretty an abstract question to think about overall. So we wanted some quantitative data to get behind that. So one way to do it is to measure grazing patterns on our sugar kelp here. But the other main way you do it at the very end of our experiment was to dissect all of our urchins and measure their gonads, their sexual reproductive organs. And ideally, the heavier the gonad is, right, the more energy they're putting towards reproduction, the less heavy, the less energy they're putting towards reproduction. So I was grabbing calipers every day to go measure the kelp and the urchins and the crabs. And I was really excited to be a part of this research. And I believe you might see this year publication, the PhD student I was working for, Nicole Peckham, come out this year. So we'll really see how that all ties together. I'm super excited to see it. And I want to give a huge thank you to the Kimbrough Lab for enabling me to do this research and really jumpstarting my career in ecology. Super passionate about that. And also a huge, huge thank you to the Halberstadt Family Summer Internship Program. It is such a blessing as a researcher to be funded for the research you love. It enables us to put more of this awesome research out into the world. So I want to give a huge thank you. And I want to pass off my microphone to Mariana. Thank you all.
All right. Hi, everyone. My name is Mariana Kalbach. I'm a third year studying environmental and sustainability sciences with a minor in public health here at Northeastern. Outside of the classroom, I do a lot of theater stuff on stage and off stage. I'm also a Husky ambassador, doing various outdoor clubs on campus, all the good stuff. But today I'd like to talk a little bit about the research I'm participating in during my co-op currently in the Lauderhouse Lab. And right now at the Lauderhouse Lab, we are studying primarily for my co-op the relative contributions of plasticity, local adaptation, and seasonal adaptive tracking in fine-tuning key overwintering traits in Drosophila suzuki'i, which is an invasive crop pest and is colloquially known as spotted wing Drosophila or SWD. So that's how I'll be referring to it from now on. So basically, you might know Drosophila as a model study research. If you're model study organism, if you're a researcher, but if you're anyone else, you know them as a fruit fly. That's really annoying in your kitchen, goes for your rotting fruit, makes a mess, But the interesting thing about SWD is that they are able to go and they have a little serrated egg laying piece, so they can lay eggs in hard or even like hard ripening or unripe fruit, which happens while it's in the field. And this is super harmful, not only for consumers, but primarily for farmers who are growing these fruits and they're making a livelihood off of that. And overall, the economy is really hurting from the amount of damage that we're seeing from this pest. It's an invasive pest. It entered the United States in 2008, and it's rapidly expanded. And one of the reasons we're so interested in researching it is because we want to understand how it's able to adapt and move and thrive in so many model environments as an invader. Now, we're able to do this research in collaboration with the University of Vermont and the University of Kentucky, and we're all kind of working on different parts of that project. At University of Vermont and Kentucky, they're doing primarily genetics and experimental research, working on that. But here at Northeastern, we're focused more heavily on creating simulations to basically test these different factors, like I said before, local adaptation, seasonal adaptive tracking, and plasticity, to understand what exactly it is about Drosophila suzuki'i that make them able to survive in novel environments, especially with climate change as the seasons get increasingly warmer, range boundaries shift, and for an invasive species, how they're able to move increasingly northward into increasingly differing areas. Oh yeah, I forgot to point out the damage up there. But since I started on my co-op, I started in Vermont at the University of Vermont in the Nunez lab. And while I was there, I helped do a lot of the field work. I went out in the field and I did a lot of collections of specimens. That was really valuable. I got to speak with a lot of farmers, and we brought those back to the lab for a lot of population counts. So as you can see, these are graphs of SWD on 4 farms from 2024. So we got to get a kind of like good scope of what those seasonal populations look like over time. But I also did a lot of outreach work while I was there. So that's mainly the stuff you can see on the left side of the slide. I created a lot of informational materials for public outreach, which is something I'm really passionate about as a scientist. For example, that poster is a sign that I made to put next to the little traps we put on farms, just to let people know, hey, this is the research we're doing, trying to explain it in terms that people can understand and hopefully get them interested in learning more. I created a little page on the website, as well as some educational materials that I presented to kids at a World's Fair recently. It's just been super great to interact not only with farmers who are intimately aware with the damage that SWD causes, as well as people that have never even heard of the organism before. And now that I'm back at Northeastern, a lot of my work has shifted to focus primarily on data analysis work. First of all, I'm helping to create an app using AppSheet, which is going to be helpful in streamlining our Drosophila research, our data collection and our sorting collection, our data collection data and sorting data. It just helps us to make it all streamlined and make it easier for analysis down the line. We're in the phase of testing that out and kind of getting feedback on what works, what doesn't work. How could we make it easy for people inputting data and actually being in the field to understand, like, wow, I'm gonna like run that back. Because I was in the field, it's really helpful for me to know what people actually want from that app, what people would want to use it for, what functions they want. All that is to say, been working on making that as accessible and as easy as possible so that hopefully down the line, our data is as streamlined as possible. And in addition to that, I'm also helping to work towards integrating some NASA weather data into our research. We want to understand how Suzuki'i are able to thrive in different weather conditions and how they're affected by different weather conditions, especially due to climate change. And with that, I've been downloading NASA data, historical data, and comparing that in coding software to our population data just to create a cross correlation analysis and see about how Drosophila suzuki'i are affected by weather changes. And yeah, it's been really great to have all these experiences as a researcher. Before my co-op, I'd actually never been in a research lab before. And with the support of my PI, Dr. Katie Lauderhouse, and everyone at UVM and our collaborators at University of Kentucky, I feel like I'm learning a lot about the research process. I hope to go into agroecology post-grad, so learning about pest management has been super valuable. And not only have the skills that I have learned been super helpful in kind of pointing me in the future directions for jobs and such, making me prepared in that area, I also feel like I know a lot more about what I want for my future. And yeah, I'd like to extend a thank you to my PI, Dr. Katie Lauderhouse, who is here. Thank you for coming. Dr. Joaquin Nunez at UVM, as well as the generous financial support from NSF, who we have a grant from for our project, as well as the Elephant Works Foundation Endowed Research Co-op Fund, who has made this experience possible for me. And with that, I'd like to invite our next speaker up.
Hi, everybody. I first want to shout out Ben. I love Mass Gen so much. Big fan. I'm running for their Boston Marathon team this year. So, anyways, my name's Gabrielle. You can call me Elle if that's easier. I'm a third year behavioral neuroscience student here at Northeastern with a minor in data science. My research experience has spanned a lot of different realms of neuroscience. I came in knowing I wanted to do neuroscience. I came in having no idea what kind of research I wanted to do. That's kind of my journey here. Currently, I'm on co-op at AbbVie. So if you know Botox, if you know Skyrizi, if you know Humira, that's that company. So right now, I'm part of their circuit biology and in vivo pharmacology group. I'm doing a lot of preclinical research on schizophrenia at the moment. Highly recommend. I love it so much. After that, I have another co-op. I'm hoping to go back to AbbVie. If not, we'll see where I end up. But I'm going to be applying to PhDs in neuroscience. So that's where I'll be. But my research presentation is a little different. Mine kind of spans my development as a researcher since coming to Northeastern rather than one project in particular. So I'm excited to share that with you all. My first year I started in Oakland, so I had the privilege of doing research at UC Davis. That's not pictured here. With a professor I had on the Oakland campus. From there, I knew I also was really interested in DEI in general, so that's a big passion of mine, and I integrated that into doing psycholinguistics research with a professor out in Oakland, which I received 2 base camp grants from, which is an internal Northeastern grant for research. So I did that my freshman year. Coming to Boston, I was really nervous about coming into a much bigger campus with a much larger student population and how competitive research would be here. I applied and was so happy and appreciative to be selected for the Dean's Research Grant, which I got for the Civ Lab for summer 1, summer 2, fall 24, and spring 25. So 4 different semesters of this grant, and it really changed the entire path of my research. This was my first experience working in vivo with any animal model, which is what I'm still doing. So for the Civ Lab, I did a lot of genotyping zebrafish for a genetic syndrome called 16p-del or 16p11.2 deletion syndrome, which is related to other things like autism, schizophrenia, and other neuropsychiatric disorders. From there, I did a study on social behaviors with our lovely MC Dante, who's here tonight, and shout out Brandon in the back is our postdoc from that lab. So he was a great help getting us going with that project. So that's what we did for the Civ Lab with our former Dean Civ of the College of Science, who's no longer at Northeastern. From there, I started working in the Shansky Lab. The Shansky Lab does a lot of neuroanatomy and behavioral neuroscience work, working with mice and rats. And this here is the first time I did a brain collection. So happy, so proud. You can call me weird for it. I was so happy. So there I look at a lot of fear responses, doing fear potentiated startle, fear conditioning, fear extinction, things like that. Again, my first really big experience with animals started the Civ Lab, gave me the motivation and the guts to pursue something bigger with rodent models. From there, I was selected as a highlighted student at our RISE presentation. For any of you who don't know what RISE is, it's a internal research conference here at Northeastern where students, I think there was 500 students last time who got to present their research. I had 2 posters up, and it was really cool just to network and meet new people and talk about how my research has spanned from linguistics to behavior in fish to neuroanatomy to biochemistry and different things like that. So that's kind of what all my Northeastern research has looked like. I also received the Russell Women in Science Leadership Program scholarship that was used towards the Civ Lab as well last spring semester, which has been so amazing. And with all of those funds and my PIs, Aaron Canale, Priya Shimpi Driscoll, Rebecca Shansky, and Dean Hazel Civ, I have been able to get my dream job as my first co-op, which is more than I could ever ask for. So the donations, the funds, and everything, all of these scholarships and grants really do allow us to chase our dreams. It's kind of, I'm sorry, I'm so excited. I love what I do. So thank you. And our next presenter is Wenchuan. Sorry.
Thank you. Hi everyone, my name is Wen Xuan, and I'm from Beijing. And I'm super grateful today because as an international student who done the second co-op in the College of Science, I'm really grateful for this very hard, especially hard period of all the researchers in worldwide that have a funding. And I'm super grateful that I received the Schaefer Scholarship Research Fund by the Dr. Andrew Schaefer, who is MD PhD at the Cornell Medicine. And so just introduce myself. I'm a senior year at Northeastern, and I'm studying cell and molecular at the Dr. James Monahan Lab. So basically, so this is my second co-op. My first co-op is at Mass Year, where I serve as ophthalmics assistant at the retina department. I remember 8 hours a day, took like 100 patients, the retina using OCT, Optos, and all of the machines, like just took the patients like retina. And that is the starting point that I really like an entry point that I know I interested in wanted to dig more into like why we have AMD, for example, why we have retinitis pigmentosa. So at I done my first co-op at the Messenger Retina department at 24, and then I just having a grateful opportunity shadowing Dr. John Miller at the Messenger Retina department. He's retina imaging lab. He allowed me to be there with the summer intern, and where I continuously to contribute in the clinical lab in the retina. So where we testing different system and the data in the newly FDA approved retina imaging, like the OCT, the advanced model of the OCT, basically in the clinical trials, and where we took like gathering the data and the retina pathologies, and we analyzing the data. And then I joined the Dr. James Monahan lab after I took the research scientist Tim Dior. He's leading the stem cell regeneration. By the way, it's a really great class, and where I actually get in touch with the regenerations, the stem cell regenerations in the field. And I, after his class, I basically sent a bunch of the email to James, who is also the chair of the department, and he didn't respond. I know he's a busy guy. And then I came to the lab. I found another PhD student whose name is Melissa. She sit at the opening of the lab, the door. I knocked the door. I introduced myself. I was like, I'm Wen Xuan. I'm really interested in the regenerations in axolotl, and and she's very kind. She's like, James will be there. Like James will be here like this week, Friday. You can just like try out. And then I met with James. I asked for the if I can join the lab, and James would, maybe saw my passion and allow me to join the lab. And then I applied for the co-op because I want to continue to doing, retina regenerations in axolotl because I was amazed by their, the ability to regenerate, their limbs, their retina, their jaw, and their lung, basically the main focus on our lab. So I was, so in our project, we aims to identify what actually we hypothesize that they have the fully ability to regenerate, to grow back their retina after complete surgery removal. So we want to investigate. So I first want to shout out my PhD mentor, Nicole. And so the first question we want to investigate is whether or not they can fully regenerate back the retina after complete retina removal. And the second things we want to investigate is if they can regenerate the retina, what molecular factors, what molecular mechanism drive out throughout the whole process they can like contribute to their regeneration through all the regenerations, the factors. So basically currently there are two main reservoir that contribute to the retina regeneration in amphibians. The first is the CMZ zone, it's right here. And second is the RPE. So basically, we are mainly focusing on the RPE. RPE is the last layer of our retina. So this is a cross section of our eye. So this is the cornea, this is the lens, and so this layer is the retina. Think about our retina is the film. So the light spin into the cornea, and our retina is the main structure in the eye under the back layer of the eye that interprets those images, that interprets those signal and transfer into the electrical signal that, especially in the macular area, the retina would take information like the motion, the color, the shape of all the information we can see, and then transfer into the electrical signal, send through the axon and send it to the occipital lobe in back of our head. And our head is a computer. So that's why there's so many diseases that leading that retina damage. If all retina damage is the first, like, mainly the causing the blindness in the worldwide. So that's why I'm very passionate. And also my grandma have the white AMD, unfortunately. So that's why it's the main reason why I'm so passionate about of the retina regeneration, especially in this. This is a axolotl. This is our lab. Axolotl. This is a pictures I found in the Google, and they're very cute. But the only thing I hate them is I have to change the water of them. I have to scrub the tank. I have to transfer them into a container, and I have to scrub them with a baking soda. And then but it is a fun part. And my lab member said I'm always put my earphone on, listen to the MCAT podcast, and then like just leave me alone and scrubbing my axolotl tank every day. So here is the two image we got. By the way, there's all control. So that's I just want to give over wide. This is the molecular structure of the retina in the axolotl. So the star, so the black layer is the RPE layers. This is what we mainly focuses on. We highly hypothesize that this is the reservoir for driving the retina regenerations in the axolotl. So what my independent project now going on is I take up the RPE and I culture them in a cell plate. So with my mentor, we're focusing on the vivo stuff, but we're also focusing on vitro. So I basically want to see how the patterns with like culturing the RPE, how they can contribute in the vitro conditions to grow back to the retina, and what is the process. Are they going in from the bottom layer to the top? So there's a hypothesis that they grow back to the retina from the bottom to up, but there's no evidence that in axolotl they can like in vitro they can fully regenerate. So that's what I'm mainly focusing on. So, and this is the cell membrane staining, cytoliner staining of the RPE, the retina. So RPE is at this layer. So yeah, so that's what I trying to do through my co-op. So this is my 4th month in the Monahan lab, and we just took some bunch of pictures of my cell culture and have some exciting things, and we see some, like, I'm not confident, but we see some neural-like structures of my RPE cells. So yeah, kind of very excited about and looking forward. And I know it's really hard for the research is that you keep fail and you keep, have the result that maybe not that ideal, but it's just also very excited that you see, you find new things. And yeah, I think that's the fun part of doing research. So I hope, I really want to thank Dr. James Monahan and also Dr. Schaefer, who is the funding of the medical research scholarship, and also my undergrad, Sonia, in the lab. So yeah, thank you. I would pass the microphone to Emma. Hi, everyone. My name is Emma Fitzgerald. I'm a fourth year cell molecular biology major with a psychology minor here at Northeastern University. I grew up in San Diego, California, which is kind of where my love for marine species began. I don't know if any of you have ever been to the Birch Aquarium, but it's awesome and I highly recommend. But nowadays, I'm more interested in the way that research on marine organisms can be used as an insight into human health and disease. So during my time at Northeastern, I've had 2 co-ops. I was a medical assistant at Beth Israel Deaconess Medical Center in Longwood, and now I'm a research assistant at the Ocean Genome Legacy Center in Nahant at the Marine Science Center. Outside of academics, I am the nutrition officer for the Empower Women's Weightlifting Club, and I also enjoy tutoring both my peers here and high schoolers and middle schoolers, though not with some of the new jokes that they like to make. It makes things very difficult to keep them on track. My research I'll get into a little bit on the next slide, but as for my future plans, I'm graduating in May and I'm currently applying to medical school. The research that I do at OGL, the Ocean Genome Legacy Center, centers around evaluating methods for long-term DNA preservation. And so OGL, the Ocean Genome Legacy Center, is home to over 30,000 DNA samples, which is a lot of samples. So it kind of focuses on investigating ways to improve DNA preservatives and overall maximizing sample integrity. And the research that is done at OGL is super important for understanding the biological diversity of our planet's marine life. And it also provides a resource for other researchers to obtain information to use it in incredibly new and exciting directions. So my student project that I focus on involves looking at the effects of different DNA preservatives on long-term storage of various tissue samples. So recently I've dissected sea cucumbers and green crabs, and I'm going to do oysters pretty soon, and that is stored for up to a year at room temperature and then extracted and looked at what that DNA looks like. So the overarching goal of this is to prevent tissue degradation and improve the yield of high quality DNA. And high quality DNA is incredibly important for downstream research application like PCR and genomic sequencing. So a lot of this is to help field workers and help research that happens in the future. So in order to visualize high quality DNA, we usually run gels, which are in the upper right-hand corner. And this is actually from a previous paper by OGL, but what I want to draw your attention to is under the E section, there are those larger, darker pieces of DNA. That is the high quality DNA we're looking for. And in the ETOH ethanol section is the smears, which is not what we're looking for. So we're really trying to find those larger, dark bands of DNA. And yeah, with that brief overview of the research that I do, I'd like to give a huge thank you to the Donald Combe Research Scholarship, which funds the student position, as well as my PI, Dan Distel, and my lab managers, Rosie and Hannah, for their positivity and direction. So thank you for listening to me today. I'd like to introduce our next speaker, Ananya.
Hi, I'm Ananya. I'm a second year student studying behavioral neuroscience and data science. In my first year, I was part of the Global Scholars Program. So I was in Oakland in my first semester, which is actually where I first met my future PI with the project that I'll be talking about and became familiar with that lab's work and spent my second semester in London. During my first year, I became involved in the Undergraduate Research Club, Girls Who Code, and I competed in the Ideathon with my team. We got first place. And then now that I'm in Boston, I've also joined a few other clubs, Neurons, the Women's Research Engagement Network, and the Research Immerse program. I will be talking about a project that I did this summer as part of the Barrett program, where we were demonstrating a fear response to alternating current in C. elegans, which is part of the Young Lab in Oakland. And now that I'm in Boston, I've joined the Chung Lab, where I'm learning about neuron regeneration and going to develop a project there. And my future plans, I will have my first co-op next year in the fall, and hopefully continue along this research track, get my PhD, etc. Okay, so the project that I joined and worked on didn't really exist when I joined the lab. So we were really developing it in the earliest stages. Already in the past, the professor, Dr. Young, he had observed during these ZAP assays that if you put these little worms on an agar plate and they have little lawns where there's food, they mostly just tend to stay on the food lawns. They're pretty happy there. They have what they need. But if you run electricity through these plates, then they run away from their food lawns. And also, interestingly, once you turn off the electricity, they just keep running for a good few minutes afterwards, which kind of signals that they're not just running as like a physiological response to the alternating current, but because there's something like psychologically going on, they're not enjoying the experience of being electrocuted. They're scared, they're panicking, and when you watch these videos of them kind of running off, it really does look like they're running in panic. So that kind of raises the question, like, is this a response due to fear, or is it like a physiological basis? What's really going on there? So in order to design that experiment, we based it loosely off of a classical conditioning model, which is something some of you may have heard of. You might be familiar with Ivan Pavlov and his dogs that you train to associate different stimuli, and you can kind of learn a lot about that underlying behavior through their learning. So I have a diagram here kind of explaining our thought process. So these worms, C. elegans, they are all attracted to this odor that we knew about. And so we were like, this is a stimulus. We know how they respond to it. It's very reliable. And also we know that if you just put them on a plate, you put the odor down, they're going to run towards the odor. That's like they end up there. This is like well established. And so that was a good piece of information we already had. And then we're assuming that, or we're trying to understand if the zap stimulus of running the electricity through their environment is a negative experience, is a negative stimulus. And so if this is true, then we can get these predictable behaviors out of them. So if you pair together the electrical stimulus and the odor that they like, in theory, if it is true that the stimulus is negative, then they will eventually learn to associate these two odors together, and they will stop liking the odor so much. Once they sort of decide that, oh, like this odor, every time we smell it, we also get electrocuted, don't like that, we're like not going to run towards the odor anymore because we don't really want to enjoy that experience. So this is what a little snippet of what the data collection looks like. You can see the plates and the copper plates helping to facilitate the alternating current. And over here is what the chemotaxis plate looks like. Basically what that means is there's little dots of odor on those X's on one side, and then we just have ethanol on the other side. So that's our control. We put the worms down and we can see how attracted they are to that odor. So after a whole process of training them to associate the odor and electricity together, we can test their attraction to the odor. And what are our results? So over the period of the summer, we were just designing this experiment, so we didn't get like final data, but this is what our preliminary results look like. We have worms that have never been put through anything. They've been living very comfortable lives, and then we just test their attraction. It's very consistent. They have a high attraction to the odor. Then there's worms that have been exposed to the odor a bunch of times in their life. We're going to see maybe that they won't be as attracted to it because it's not as new and exciting anymore, but they still have a very high attraction to the odor. Worms that have been electrocuted but have never been in the presence of the odor before, they also have a high attraction to the odor. And the main group that we're interested in is the group of worms that has had the training of associating the electricity with the odor. So our predicted results is that they would have a lower chemotaxis index. They would not be as attracted to the odor. But so far in our very early experiments, they have pretty strong attraction to the odor still. Um, However, this is not disheartening. One way to interpret it is that, oh, they're not, this isn't a fear response, but since this was only over the period of the summer, it's mainly just like a data that tells us that we still need to refine our protocol. We need to figure out how to make this association stronger, figuring out, like the very technical details of that. And so um, look forward to hearing more from Dr. Young about how they figure that out in the future. But I was really happy to have this as one of my first research experiences, and very lucky that I had only had one research experience in the past when I was in high school, and it was very focused on like data analysis. And so this was my first experience in a wet lab. And since this lab had no grad students or postdocs, all of the work was done by undergrads. So this was a really big learning opportunity where I got to really dive into understanding experimental design and literature review and doing experiments, everything in a really, head first manner, So that was a really incredible opportunity, and I'm very grateful for the Jill Barrett Research Program for making that possible. I'd also like to extend thanks to Dr. Jared Young, the PI, and my lab partner during the summer, Jillian Garcia, who was another Barrett scholar, as well as all the other members of the Young Lab who were also supporting the work, as well as the Kimura Lab, which is a lab in Japan that we kind of collaborate with, and also have done some of the ZAP assays that we were building this work off of. So I'd like to call our next speaker, Melise.
Hi, everyone. I am Melissa Arroyo. I'm a second year behavioral neuroscience major here at Northeastern University. And I'm really excited because I'm going to be starting my first ever spring co op. I'm at BMC as a PT aide. So to get into some of the stuff I'm involved with here on campus, I'm in the National Society of Black Women in Medicine, and I'm in the Dream Orchard Gardens mentoring program for the local youth. And I believe through those experiences, I really developed a keen interest in adolescent development and kind of why adolescents act the way they do, which is what led me to the lab that I'm in now, which is the Learning and Brain Development Lab here at Northeastern University, where I work under Dr. Juliet Davidow and alongside Nick Cathius. And in our lab, we use non-invasive brain imaging and computational models to kind of analyze how adolescents learn and develop as they age. And the specific research that I worked in, we took that info and then we applied it to learning, memory, and music. So that's what I'm going to be talking about today. And we had hypothesized that adolescents who are more reward sensitive would actually show less adjustment to occasional errors and more adjustment and stronger memory towards rewarding outcomes. And it's a really unique and niche topic to me because it takes something so universal, such as music, and it applies it to learning and brain development for children. There's this idea called the reminiscence bump, and it basically implies that adults are more likely and more interested in music from their adolescence. And it kind of relates to emotional and self-defining memories at that time. So that kind of builds our gap because we take something that's been previously defined as meaningful, such as music, and we apply it to how it can benefit adolescents in relation to learning and memory. So right now we're in the data collection phase, so we're still interviewing participants to further develop our findings. This is a very important topic to me because it kind of takes in emotion, cognition, and development and kind of ties it all together. And we take something so niche like music and kind of apply it to its comparison in relation to like points or money. And we found that personally meaningful stimuli, such as music, are actually much more effective and more able to be broadly implied along adolescents and adults. So in our lab, Dr. Juliet Davidow, our PI, had found that neurocognition is actually adaptive, which implies that adolescents aren't bad decision makers, but quite the opposite, that they're learning and developing based on the stimuli and environment around them. And that's where music came in hand because it is a stimuli that we've learned is so influential on adolescence and development. And that's what we applied it to see how music can manipulate learning and development. How the task works is you listen to different musics, you make different choices based on the music, and then there's a memory task after. And we did that range from adults to adolescents and see the differences in how they reacted to the stimuli and the feedback that they gained. It really inspires me because it kind of takes a naturally engaging stimuli and something so naturally versatile to the world and then applies it to learning. And me wanting to be someone that's going into medicine, specifically a pediatric surgeon, I can see how this can apply in the clinical space when applying and teaching kids to regenerate and kind of grow clinically. So this research kind of translates into a very impactful way for me because working with Juliet and Nick, they kind of shown me that research can transform into clinical care. I kind of wanted to take this and take something in my own world, like when I apply to medical school, when I go into, and this is something I'm really proud about and hope to take alongside with me, if it be through education or therapy or medicine. I really hope to apply what I've learned here and apply it to my future endeavors in relation to in relation to clinical care and how adolescents grow and develop. And yeah, I just want to take a second to thank Dr. Juliet Davidow and Nick Cathius for really guiding me through this experience because this is my first research lab and I'm really excited to be here for the next 3 years. And I would love to thank the COS Dean Research Grant for kind of funding me throughout this whole experience in this lab. Yeah, thank you. I'd like to thank you for time.
Hi everyone, my name is Kai Godwani. I'm a fifth year majoring in marine biology and minoring in visual art. I've completed 2 co-ops. The first, I worked as a cephalopod aquarius intern at the UChicago Marine Biological Laboratory. The second, I worked as a toxicology research assistant at the Woodslow Oceanographic Institute and U.S. Environmental Protection Agency. My first semester at Northeastern, I studied abroad in Dublin, Ireland through the NUN program. And when I returned, I joined the Hawaii Club. And for the past 2 semesters, I've been working as a teaching assistant for our ecology lab and a marine environmental science peer mentor. And this past summer, I had the opportunity to work under Professor Aaron Stubbins as a Jill Barrett Research Fellow, investigating chemical digestion microplastic isolation techniques. And looking forward, I'm continuing my research as my capstone project, and I'll be graduating this December and hope to be starting a research assistant position in the spring. So, as I'm sure you all have seen firsthand or in the news and media, plastics are everywhere. And this has led researchers, including the Stubbins Lab, to develop techniques that allow us to isolate microplastics from environmental samples. So our procedure begins by collecting an environmental sample, whether that's soil, water, animal flesh, adding that to a highly basic solution, heating that in the oven overnight, which will cause the production of radical species. This will eat at and break down our organics while not being able to break down our tough plastic polymers, allowing us to vacuum filter the resulting solution and just the way the resulting plastics. And these methods worked well for our researchers, but didn't look at the effect of size on recovery, and also found that some of the plastics were being lost, suggesting that smaller plastics might be being digested alongside the organics. And as my job was to optimize these methods, I wanted to address this possible under recovery, so I focused my experiments on one plastic polymer, polyethylene terephthalate, or PET, which we would see as a plastic water bottle. I also chose size ranges that included super tiny plastic powders, as fundamental chemistry tells us that smaller things digest more readily, the same way a scoop of granulated sugar dissolves faster than a sugar cube in your tea. Following the procedure, I completed 16-hour PET digestions and found that PET did in fact digest at all of our size ranges and more in the smaller sizes. But as researchers, we wanted to take this a step further and model the kinetics of the reaction. So we identified the shrinking sphere model as a representative model for our process that takes into account a suite of assumptions and particle parameters to derive a linearized equation that only requires we measure the fraction of plastic dissolved and time elapsed to calculate dissolution rate. So I completed these experiments again, this time stopping the digestions at different time points. So I had time series data set and inputting that data into our formula and plotting it. As you can see in the top right, we have strong linear fits at all 4 of our size ranges, validating our hypothesis that this pattern of size on digestion rate is due to surface area to volume ratio. And taking this further, we can take our dissolution rates from each of our slopes and plot it against average particle size, which shows an inverse relationship between digestion rate and size, validating that smaller things do in fact dissolve quicker and that this is due to surface area. And finally, bringing everything together, I identified that existing chemical digestion techniques might be systematically underestimating PET contamination in environmental samples. And looking forward, I hope to use the same model and framework on different plastic polymers, as well as samples from the environment to inform us on how we can refine these methods. And finally, I'd like to give a huge thank you to the Jill Barrett Foundation, Aaron Stubbins, and the Stubbins Lab. None of this would have been possible without them. And I'd like to bring to the stage our last researcher, Jay.
Hello, my name is Chunghyun. I go by Jay, and I'm a fourth year biomedical physics student here at Northeastern with a minor in mathematics. And some of the student activities I'm involved in is I'm a secretary at Society of Physics Students. I'm also involved in Sigma Xi Research Honor Society, where I'm also part of the Research Immerse program as a mentor. And I'm also a COS peer advisor, and I am currently a student researcher at Spring Lab, where I finished my second co-op. And the two major research projects that I've done over my two co-ops is the first one I've done was 5-aminolevulinic acid-based photodynamic therapy for enhancing immunogenicity in mouse oral carcinoma models and in vivo models, which I've done under Dr. Hassan Lab at Harvard Medical School at Mass General Hospital. And the most recent co-op that I've done was on treating high-grade serous ovarian cancer using photodynamic therapy and chemotherapy and characterizing its treatment responses. And for the future, I'm currently applying for PhD programs in related fields of biophysics and immunology. Yeah, so to quickly go over the general idea and direction of my project, a quick background on photodynamic therapy. Photodynamic therapy is a form of cancer treatment where we administer photosensitizers. We are light activated chemicals into a tumor region. And once these photosensitizers are accumulated in the tumor region, we are able to shine those regions with a specific wavelength of light that reacts to the specific photosensitizers we use. And upon irradiation, these photosensitizers create radicals and destroy the cells that have accumulated the photosensitizers. And if you look at figures C and D, these are the two cell lines that I worked on. And as you can see, the bottom axis is the light dose. And once it reaches a light dose of around 15 to 22 joules per centimeter square, you can see there's almost complete killing of the cells. And we can see that from these graphs, both cell lines are very sensitive to photodynamic therapy. And the reason why we're able to say that is because the light dose range where we consider a high dose would be 50 and above. And because these cell lines are almost completely dead, around 22 joules per centimeter squared, we can conclude that these are very sensitive. And looking at graphs E and F, these are flow cytometry results based on the cell surface markers that we have identified from running proteomics. And we are able to see that two different cell lines which are both ovarian cancer related cancer cells, have different biomarkers which we are able to now use to create targeted therapy which can be more selective and more effective and improve our treatment outcome. And with these information, the future direction of this project is to utilize these results to create a multiple cell surface utilize these multiple cell surface markers for multi-targeted photodynamic therapy, where would be able to target specific markers of each cell, and instead of only targeting one part of the cell by multiple sites of the cell, we are able to deal with the heterogeneity of each cell line, which can also improve treatment outcome. And using these surface markers and having developed a targeted photodynamic therapy, we're able to further it into a photoimmunotherapy where we are able to activate the immune system upon irradiation, and introducing PBMCs, which are immune cells, will also help with immune activation. And with that, I would like to thank the Sanctus Research Co-op Fellowship for funding my research, and my grad students who I've been working on, who's now graduating in Nepal, Sudeep, and my PI, Professor Brian Spring, for helping me along the way. Thank you. And I'd like to pass the program back to our MC, Dante.
Thank you. Let's give one more round of applause for all of our student researchers, mentors, would not be possible without anybody in this room. I would like to congratulate the student researchers and say thank you for all being here, taking the time out of your night to come celebrate everybody. And I hope you enjoyed your night and be ready for dessert. And want to give a special thank you to all the people listed on here: the College of Science Dean's Fund, Jill Barrett Research Scholarship, DeSantis Research Co-op Fellowship, Haverstadt Family Summer Internship, Robert E. Matt Scholarship, Andrew I. Schaefer Co-op Research Fund, Shots Research Co-op Fund. I'm sorry, I mispronounced that. The Paul and Grace Ward Martinez Research Co-op Fund, the AJC Merit Research Scholarship Fund, Donacon Research Co-op Fund, Russell Women in Science Leadership Program, Elfin Works Foundations Endowed Research Co-op Fund, Marine Science Center Co-op Fund, and the Professor Paul Voros Scholarship Fund. Without these funding opportunities, many of us would not be able to participate in research and engage in science. So thank you so much. And dessert shall be in the back, but please, before you get up, take a moment and provide us with feedback. Please scan the QR code and provide feedback for us. Thank you all.